Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And remember that you can find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Google Podcasts, Pandora, TuneIn Radio, Stitcher, Overcast, Good Pods. So, wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Twitter as Let's Talk Micro 1, on Instagram as Let's Talk Micro, no apostrophe, and on LinkedIn as Luis Plaza. So go ahead and follow, subscribe to the podcast, leave a review. That's always good for the podcast. So if you listen to an episode and you like it, go ahead and rate the podcast and leave a review. And also you can hit me, via social media. I have an email, let's talk micro at outlook.com. So any suggestions, any feedback, any podcast topics, they are always welcome and appreciated. And if you haven't listened to the previous episode, go ahead and do so. It was a great episode with Dr. Paul Hergenrother from the University of Illinois, and he came to the podcast to talk about fabimycin, which is an antibiotic candidate that has shown, activity against some common gram-negative organisms that are seen in infection. So go ahead and check out the episode. Find out which organisms fabimycin has shown activity against, and find out what is fabimycin. How does it work? What is its mechanism of action? So find out all this information by listening to the episode. It was a great episode, very informative. So go ahead and check it out. So on today's episode, we are actually adding a little bit of culture to Let's Talk Micro. So we typically talk about the clinical aspect of micro. We talk about medical laboratory sciences. So today we are bringing in a little history. It's definitely related to what we do at work, but it's always good to learn about this stuff. To get educated. Definitely, I'm all for learning, all for education. So we are bringing a little bit of history. And in this episode, we have two guests. We have Philip Slavin from the University of Stirling in the UK, and Maria Spyrou from the University of Tübingen in Germany. She's from the Institute for Archaeological Sciences. So, great guests, they brought some great information to the podcast, and they came in to discuss an article titled The Source of the Black Death in 14th Century Central Eurasia. It is definitely a great article. And, all of us basically in school, going through school, we have heard about the plague, right? The plague pandemic. So we have heard about it. We have learned about it in history class, definitely in the lab in microbiology. We learn about Yersinia pestis, and we learn about the plague. I mean, we don't get to work with this organism, thankfully. At least, in our part of the world, we don't see it. I mean, we do train to recognize it, but we definitely learn about it. So they came to the podcast, and they talked about how, there was some ancient DNA that was obtained from 7 individuals from 2 cemeteries, and then with this information, there were 2 Yersinia pestis genomes reconstructed, and they were identified as a common ancestor before there was a divergence. So in essence, this data supports a source, an early source of the second plague pandemic in Central Eurasia. So they did a great job of explaining it. So let's go ahead and listen to the episode. So on today's episode, we are talking about the plague. Even you have heard about it in school, university. As laboratorians, as clinical microbiologists, we are trained to recognize it every year to make sure we are able to detect it in the lab. So today here I have 2 guests to discuss an article titled The Source of the Black Death in 14th Century Central Eurasia. This was published on June 15th of this year on the Nature Journal. So with me, I have Drs. Philip Slavin and Maria Spyrou. Doctors, welcome to Let's Talk Micro.
Thank you very much, Louis. It's great to be here.
Thank you so much. It's great to be here.
My pleasure. So let's go ahead and start with an introduction.
So I'm Phil Slavin. I'm an associate professor of environmental and medieval history at the University of Stirling. I've been interested in the history of plague and Black Death in particular ever since I remember myself as an undergraduate student many years ago, but not too many years ago. And I remember one of my first experiences as a student in those huge intro classes. I remember my professor telling us that there are so many different theories about the origins of the Black Death. And one of his conclusions was that there's absolutely no way we'll ever be able to solve this enigma of the origins of the Black Death. So I really hated this statement. And I guess I've been living ever since to disprove that.
And my name is Maria Spyrou. I'm a postdoctoral scientist at the University of Tübingen. And yeah, also for me, when I started my PhD in 2014 in archaeological sciences, but my background and my training is more in biomedical sciences and medical microbiology. And so I've always wanted to be able to, or was hoping to be able to pair this interest of mine in medical sciences and microbiology with also my interest in history. And this was a fantastic opportunity. And so since 2014, I've been part of this big expanding field of ancient DNA, and specifically the one of ancient pathogen genomics. And it's been great to see all the developments that have been coming out over the last few years on the history of plague, which I have focused the majority of my career at so far.
So let's go ahead and do a historical overview. Let's talk about, so as far as the first plague, what was the location and the population affected?
Right. So the first pandemic, the origins and the progression of the spread is still a huge mystery to all of us. We don't really know where it started. What we know is that it arrived in Egypt in one of the local ports in the year 541. We don't really know where it came from. There are lots of different theories. One theory holds it started in Central Asia. The other theory holds it started somewhere in sub-Saharan Africa, maybe in Ethiopia. We don't really know where it started. Unfortunately, the surviving sources are not very good in telling us actually about the extent of the crisis that it created. There's a recent hypothesis suggesting that actually, contrary to the second plague pandemic, it wasn't actually as devastating when it arrived. And I'm sure that in due course, new ancient DNA work will be able to answer some of those pressing questions. But we're in much better position actually to talk about the second pandemic, because we have really fantastic sources, both historical sources and ancient DNA. But I wonder if Maria has something to add to that.
One very interesting aspect about the first plague pandemic is that ancient DNA has really been able to sort of meaningfully contribute to our understanding of the pandemic, because we've been able to generate data sets, and I'm talking about data sets of the Yersinia pestis DNA from regions where we don't have historical evidence or historical records that describe the pandemic. And so this is a really great case for seeing how history can really pair up with genetics to really give a perhaps more complete picture of what was going on at that time.
Okay. So then let's go ahead and let's talk about the second plague pandemic as far as the location and population affected.
Right. So the historical origins of the second plague pandemic, beginning with the Black Death, has been one of the biggest mysteries in history. And that's something that has been puzzled historians and archaeologists for several centuries now. In fact, the contemporaries of the Black Death, people who actually witnessed that in the early 14th century, had no clue where it came from. And this is something which kind of, created those foundations for this curiosity and the inquiry about the origins. So we can, broadly speaking, divide main hypothesis, the most major hypothesis, into four different groups. So there was a school of thought that blamed China as the country of origin. There was another school of thought that thought it was started somewhere in Central Asia. Then you have people, both historians and the scientists, who thought that it started somewhere in the somewhere between the Black Sea and the Caspian Sea, so between Crimea and Caucasus. And then you had people who thought it may have started somewhere else, maybe in India, maybe in Mongolia, maybe in, even in Siberia. There was a study that suggested that. And one thing in common to those hypotheses was that they didn't really solve this long-standing controversy. They only complicated this controversy. I mean, one problem was that the first textual evidence, and I really stress the textual evidence about the outbreak of the Black Death that was known to historian, comes from 1346 in Crimea. We know about this really famous episode when you have the Genoese, trying to hold on to their to the fortress in Crimea, a place called Kafan, and on the other hand, you have the Mongol and the Tatar armies catapulting allegedly those dead bodies into the Genoese camp, and that's allegedly how this whole thing started. And the Genoese took it to ships after that and spread it to Europe, but it didn't really solve the puzzle where it started.
Now, beyond the Black Death, the second pandemic is a much wider period, spans a much wider period in history, which is not only the 14th century, mid-14th century Black Death event, but it actually spans almost 5 centuries. So from the 14th until the 19th century. And an additional point is that one of the main aspects of the breadth of the pandemic was always which regions were affected. And of course, our Eurocentric view of this pandemic has oftentimes biased us in studying mainly regions around Europe or Europe itself. But there's much more evidence that is now and research that is currently underway that suggests that there was the pandemic had a much wider impact.
So as I was reading the article, it mentions that there is an ancient genome. So I was just curious, so where was this data obtained from?
So in 2017, I came across a really interesting publication. It was a publication by a late 19th century Russian linguist or philologist, to be more precise, and he published an entire corpus, an entire data set of recently excavated tombstones from one Christian cemetery in northern Kyrgyzstan, in a place called Karadzhigaj. And I was really, really impressed by this very rich corpus. But one thing that impressed me the most was the fact that almost every single stone had been dated. So we know that there is a corpus of over 400 tombstones, and they're all dated to the period between 1248 and 1345. And I could tell right away that there was one single year where we have a remarkable spike in the annual number of burials. That was the year of 1338, namely 8 years before the arrival of plague into Crimea, the same episode that I was just discussing. And I told myself, wait a second, cannot be possibly just some sort of coincidence, must be some sort of connection there. So I started delving more and more into this corpus, and later on I published a paper, but there was no way for me to actually prove this connection between the spike in that one single year, 1338 and 1346, and the beginning of the Black Death in Europe in 1347. That's 9 years later. So I kind of convinced myself right away that without a DNA, there's absolutely no way that we can prove it. So I started hunting for the actual specimens of those people. And after much search and hunt and research and getting connected to new people that never I knew before, I finally managed to pinpoint the exact location of this collection. The samples had been picked up in early 2018, and Maria will be in much better position to actually tell you all the exciting developments that happened after that.
Yeah, so we were able to obtain specimens from 7 individuals that were found in those cemeteries in northern Kyrgyzstan. And we were quite happy by the fact that we were actually, for most of those individuals, we were actually able to associate the skeletons, this from where the specimens came from, with the individual's burial, and of course also the tombstones that were oftentimes or most of the times describing the year in which those individuals died. So we had actually now the opportunity to really test whether this 1338 event was really an epidemic of plague or it was some other pathogen. And that's actually what we did. We extracted the ancient DNA, or we extracted DNA from teeth of 7 of those individuals, and we sequenced this DNA, and then we were able to, using computational tools, to actually try and identify what microorganisms we might be able to find in those sequenced reads.
Okay, so yes, well, that's very fascinating information. So with this, this was the information that the study is about, right?
Yes, exactly. So we were able to study 7 individuals from the Karajigach archaeological site. This is a site that was excavated in the 19th century, as Phil described, but it was excavated in the 19th century, but the individuals buried at the site were dated mainly to the 14th century, with a large spike of individuals that died in the year 1338. So when we analyzed 7 individuals from that site, we actually had the opportunity to directly test the hypothesis of whether the individuals that died during this 1338 epidemic were really victims of a plague epidemic. And that's actually what we did. So we extracted a DNA from 7 teeth obtained from those individuals. We sequenced this DNA using next generation sequencing technologies, and then we were able to mine this DNA fragments that were sequenced for looking at what kind of pathogens we might be able to identify in our data sets. And one of the pathogens that came up when we were doing this type of analysis was the Yersinia pestis pathogen, which is the causative agent of plague.
Okay, thank you for that. So, as also with the article, I was reading about, there are 4 major lineages of Yersinia pestis. Can you talk more about that?
Yes. So, of course, this was not the first work that was ever dealing with ancient DNA from the second plague pandemic. There had been a number of papers prior to our study that had sequenced genomes from the second plague pandemic, including genomes associated with the Black Death. The issue was that all of those previously published genomes were from Europe, so they were not so suitable for telling us much about the source of this pandemic. Now, what we learned, though, from the generation of this data in previous studies was that the Black Death was associated with the birth of 4 major lineages in the history of Yersinia pestis. The genomes that had been previously published from the second plague pandemic period were associated with what we call branch 1. So branch 1 is associated with the second plague pandemic, but it's also associated with the third plague pandemic that began in the 19th century and spread plague across the world. And it's, for example, one of the reasons why plague is present in the United States today. Now, we also have 3 additional lineages that were born as a result of this likely of the Black Death event, which is lineage branches 2, 3, and 4. And those are lineages that are mainly identified today in wild rodent populations that are also the main hosts of Yersinia pestis in Central and Eastern Asia.
And if I could also add to that, one big difference between the branch 1 and the lineages 2, 3, and 4 is that the branch 1 had much faster and also much more diversified evolution after the Black Death. If you look on the history of lineage 3 and 4, those are, they were developing quite slowly. There are about 15 to 18 snippets between the Big Bang event and what we have today. By contrast, if we look on the evolutionary history of branch 1, where between the 13th century and the early, between the 14th century and the early 18th century, you have 90, no less than 90 snippets separating from this great polytomy or the Big Bang event and the outbreak in Marseille in the early 18th century. That's another thing that we have to account for. And also geographically speaking, as Maria already alluded, quite rightfully, branch 1 has much more diversified, much more global circulation, whereas other lineages are confined primarily to Central and Eastern Asia. So that's another thing that we have to keep in mind.
Okay. So as far as, just for the audience, so, we went through all the information, what you gathered, what you found. So just to explain, summarize everything. So how do these findings relate to the source of the second plague pandemic?
Yeah, so as we already discussed before, we were able already prior to the study to associate the Black Death with the birth or the diversification of 4 major lineages in the history of Yersinia pestis. We know that the Black Death event was associated with this birth, but it was not falling exactly on the node of this diversification. It was falling in association with branch 1, and it was specifically only 2 mutations away from the node that gave birth to those 4 lineages. So what we were able to find when we analyzed this new data set now from Central Asia was that the genomes that we could isolate from those individuals, the Yersinia pestis genomes that we could isolate from these individuals, were falling exactly on the node. So on the birth point of this major diversification. So genetically, we were able to find the source strain, if you like, for this major diversification event. Of course, geographically, that wasn't telling us necessarily where the pandemic began. And that's where our knowledge of the modern distribution of the pathogen came in. So what we wanted to do as a second step is to look at the possible areas in which this strain may have been born. In order to do that, we actually looked at all ancestral branching lineages in association to this strain. So where are all the ancestral the most closely related lineages in association with this strain found today in the world? And when we did this analysis, we were able to see that the lineages that are branching immediately ancestral to this strain are quite geographically confined and are found today in the region that spans the Tian Shan Mountains. So not very far from where we identified also the ancient strain. And that's what actually led us to propose that most likely the strain evolved locally rather than it having been introduced from a faraway place.
Okay. Is there anything else that you want to add? Thank you. This has been definitely, very educational, some fascinating information. So I want to thank both of you for taking the time to coming over to Let's Talk Micro.
Thank you very much for having us, Luis.
Thank you so much.
All right. My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoyed listening to this interview about Yersinia pestis and the plague. Some great information. As always, I enjoy sharing this with you. Continue bringing that passion to what you do. It's so important. I can't say enough. It makes you better at your job. Such great work that we do. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time, bye.